All right, welcome to week 3. Oh my gosh, it's already week 3. That's so crazy. So this week we are going to be discussing some different techniques that are used in testing within microbiology. These techniques, of course, cross over into other departments of the lab. For our purposes, though, we're always looking at it from a microbiology perspective. So we'll just get into the 2 PowerPoints that are going to go through this. Let me share my screen before I forget. So we can, all right. So we're going to start with the PowerPoint that's called Immunodiagnostics of Infectious Diseases. So essentially, these are a lot of immunological techniques, meaning things that use antibodies. Anytime the word immunological or immune is used, it's always referencing that probably an antibody-antigen reaction is happening. So that's what's kind of happening here. We have a lot of different techniques that are used in various kits and things like that that kind of go on these principles. Now, if you've taken the immunology course, this is probably going to be hopefully a review for you on some of this. If you haven't, that's okay. This is pretty much introducing you to this world of immunodiagnostics, and you'll get more of it out of immunology. So first we'll start with agglutination. First, agglutination and precipitation are very similar. They're both visible reactions of an antigen-antibody clumping together, essentially. So we can't visibly see with our eye, of course, antibodies and antigens. But when they join together and then they all join together with a bunch more, it does make it visible to the human eye in the form of like a clump or an aggregate, or in this case, maybe an agglutinant or precip. But there is a difference between the 2 terms. The key difference is in the precipitation term, it talks about a formation of a mass from previously soluble components, and that's the key term there to make up a precipitation. Agglutination is the clumping of particles that have antigens on their surface by antibodies, and they form bridges with other antigen antibodies to make it large enough. But again, the key difference is precipitation is involving soluble components. So here's kind of an example of what agglutination will look like, just to try to keep it in mind. So the Y shape is always an antibody. We usually draw it out as a Y shape. And when you get in immunology, you'll learn why it's drawn out that way, what it makes up its components. And then it will bind to antigens, which in this case are shown as green little spiky guys. So when they bind these, they can kind of start to cross-link together here. Do you see how down here they're starting to form together 2 in a row? In order to make agglutination, you have a bunch of these that kind of link up together, and that's how they become visible to our eye. And we use agglutination in a lot of different testing methods. So that is something, I mean, there will be a test that you guys will run in the micro lab this session in which you're looking for agglutination as your positive results. It's usually a positive result. There's something more specific called a latex agglutination. In this case, it's where we're using latex beads. So we bind latex beads to the antibodies, and then if they find their antigen and bind to the antigen, it becomes a visible clump, a visible aggregate with those latex beads and antigen together. And a lot of times kits will use these latex particles, and so they're using them as kind of like the antigen looking for the antibody instead. So if you think of it backwards, where we just talked about the antibody bound with the latex bead and we're looking for the antigen from a virus or something that's happening, whereas in other kits, like a good example would be pregnancy tests, we are using latex particles to pretend to kind of act like the antigen, or they're coated with the antigen, and then they're looking for their corresponding antibodies. So in the case of pregnancy, they're looking for their antibodies that are made when somebody's pregnant to determine, yes, you're pregnant. So just a kind of a note on that. There is a nice picture, because I think pictures are always helpful. So you can kind of see how they put the latex beads with the antibody there. And then if there is antigens, it will connect on. And it just allows for a really nice agglutination reaction. So here's some more of what it would look like in some tests. This letter A, I believe, is the Coagulase test that most of us use in the lab. That's the test that you guys are going to run in micro this session that will have agglutination that you look for. So you should see some clumpiness like this on the left side, whereas on the right side of that letter A, it's nothing, so that just is a negative. Or sometimes they use them in tube form, so you can see the difference there. So that's what we're looking for when we look for agglutination in different kits as a reaction. There's direct bacterial agglutination, in which they are using bacteria to detect antibodies that are directed against those pathogens. So if we think somebody has a certain infection by a bacteria, we can take a suspension of known bacteria and look to see if that person has produced antibodies against them. If they did, that's a sign that they probably had that infection. Indirect or passive hemagglutination. The word heme is going to tell us it involves red cells. So in this case, now we're looking for agglutination of red blood cells. Again, you're looking to detect antibodies that were made, and we coat the red cells with a substance, whether it's bacteria, fungi, parasites, and then we kind of look for the agglutination, again, clumping. Now, in the case when it's called indirect or passive, this gets really tricky because it's the opposite of what you want to think. If it does clump, it's a negative result here. If it doesn't clump, it's a positive. So this one is always backwards. Everything else for agglutination, when there is agglutination, it's a positive result. When there's not agglutination, it's a negative. In the case when it's called indirect or passive, it's the backward results. So like it says on this bottom bullet, it's called indirect or passive because it isn't the antigen found on the red cells, but the coded antigen that the antibody will bind to. So they already purposely coded their antibodies. And so if they do clump up, that's just something the lab did. So it's a negative reaction. If there's no clumping, that tells them, yes, there was a positive. I'm not going to get in detail. This probably is really confusing. I do not test you on this technique. I do not test you on the theory of this, because to me, this is more immunology-based, but it's just a mention. So it just goes on to explain it here with just regular hemagglutination. All right, so factors that are going to influence agglutination. So there's always two stages for agglutination to occur. The first stage is what we call sensitization. This is where your antibody will actually attach to your antigen. So they bind together. And then the second stage is that lattice formation where a bunch of them start cross-linking together to make it visible. So those are the official two stages of agglutination. Now, we can use different things like pH, temperature, incubation to try to make it so it happens quicker, so agglutination is more impacted and more pronounced, so that we can hopefully make sure that if it's going to happen, we help it. So there's different things that we can try to make sure that we really make the ideal setting for agglutination to occur. All right, so that's just showing those the first stage is where your antibody antigen is going to join together, called sensitization, and then you can see at the bottom the lattice formation, making it visible as agglutination. That's the second stage. All right, there is agglutination inhibition. Again, this is the backward results. Agglutination represents a negative reaction. No agglutination represents a positive reaction. So in this case, this one's used a lot of times with pregnancy kits with HCG hormone. If that hormone is present, it will neutralize the antibody, not allowing agglutination, but that would be a positive reaction here. If there's no HCG present, then they can bind together. The antibody and other things will bind together, and then that's a negative reaction. Flocculation is just another word for precipitation, and again, it's all about using soluble components. There's two very common precipitation or flocculation tests that we use in micro for syphilis detection, and that's VDRL and RPR. These are two screening tests that we use when we're looking for syphilis within somebody. Other types of precipitation tests are radial immunodiffusion, electrophoresis, and immunoelectrophoresis. So we'll start with radial immunodiffusion. So in this case, they're going to use an agar plate, which I show you here with little wells in it. So you can see there's little wells around the perimeter there, and they're pipetting substances into it. So these agar plates are already going to contain some sort of antibody. They'll either contain IgG, IgA, or IgM. They will fill up those little wells with a dilution or a serum of some sort. They'll incubate it, make it nice and toasty warm. And as the antigen diffuses out of that well and into the agar, it will react with the antibody in the agar. So again, if you put the patient's serum into the well, the antigen in that patient's serum is going to leak out of the well, And if it's present, it's going to make a bind with the antibody that's in there waiting for it. When it binds together, it does make a precipitation, which is visible as a ring. So the larger the ring, the more antibody that patient, the more concentrated antibody-antigen reaction that's occurring. So if you look here, like the one that looks like a kind of a V shape is a really solid ring saying this is a nice, decent reaction. Now, you can have different substances in each of these wells. It doesn't have to do the same thing in every one of these. So it just depends on what you know that you put into each of these. It will vary, and then that you'll base that on your reaction. Again, all of this is not tested on really on our quizzes. So if you're confused or lost at this point, it's just information. This is just a lot of information about different techniques that are used. They're easier to understand when you actually play with the techniques itself. So these two chapters, I really don't actually test you on in our quizzes and exams. This is more background. You'll do a discussion post to kind of gain some more information, but otherwise this is just providing you with different techniques that are out there. All right, so this one is comparing different immunoassay techniques. We have radioisotope versus enzyme techniques. So it just tells you kind of the advantages, disadvantages of it. I will say this, EIAs, enzyme techniques, immunoassays are extremely common. This is a very common one to see. We rarely will see radioisotopes, and the reason for that is a radiation hazard. So pretty much we play a lot with the enzyme immunoassay in lab. All right, so this just talks about using enzymes or radioisotopes to label your antibody. Basically what that does is when they form a reaction with an antigen, it will trigger a color reaction or fluorescence or something. Whatever that label is meant to do, it will trigger a reaction. And that will be your sign of when you see it, oh, it bound to an antigen. So that's the purpose of using a label. So this just discusses enzyme immunoassay, again, very common one to use. There's different types of enzyme immunoassays. And again, by using an enzyme label, here we're looking for a color change to tell us that there was an antigen-antibody reaction. So instead of agglutination, in this case, we're looking for color change. So this just shows you one type of how we use it. So you have your antigen already sitting in your plate. You add the serum, and in the serum is the antibody. They will bind together, and you don't know it yet. It doesn't make any clumping or anything. It just sits there, so you can't see it. But when you add a second antibody with the enzyme color on there, then it will form a color letting you know, yep, it was there. So it's great in like strep test kits use this. So if you look here, letter A is negative. You see there's no color reaction, nothing happened. Letter B shows you how the color change can work to show you a positive result. All right, you can also do the same thing with fluorescence. You can add on fluorescent labels, so of course they will fluoresce when there is an antibody there. So it just shows you that, again, there's two different methods, a direct and an indirect. I will not get into the difference right now. The indirect just uses a second or third item that isn't always in the direct one. But anyway, this is, again, very much immunology based, but because we do use strep tests, influenza test kits, there's all sorts of different kits, C. difficile, all these different test kits will use some sort of principle of immunology in that kit to look for what the antibodies that the patient has made against the infection. So again, I'm not going to put this on a quiz or exam, but it's just helpful information for now. Okay, and then we'll go over to our other PowerPoint, which is about molecular diagnostics. It's brief, and this is kind of more what your discussion is on this week. So to discuss some different molecular techniques that have really come out lately. All right, so molecular techniques are using nucleic acid hybridization. So lots of stuff with your DNA and or RNA. So whenever we think molecular, you should be thinking of DNA, RNA, things of that nature. So during the technique, one strand is going to be your target strand, and then that is the strand that we will kind of suspend. And the other strand, because remember, it's always a double-stranded DNA, Your other strand is going to be your probe, and we're going to label it so we can easily detect it. All right, so there's different types of hybridization methods to this. And maybe these terms are familiar to you. You can use different hybridization techniques called Southern blot, Northern blot, or Western blot. Southern blot is ideal for DNA stuff. Northern blot works more with RNA stuff, and Western blot works mostly with protein things. So again, you will take the target nucleic acid, you'll transfer it to a membrane, you will join the label probe to it, and then you're going to detect it based on the color or fluorescence or whatever you use for that probe. And so they use this kind of similar in the blotting technique. Western blot is a huge one for confirming HIV. It will look for a specific protein that is known to be with the HIV virus. So that's a huge one for confirmatory HIV. So it is something that we do see and work with in different labs. All right, the biggest things that are coming out right now are different PCR techniques and probe-based assays, especially for detection of bacteria quickly. So they have a GenProbe assay that's used to detect chlamydia and gonorrhea. So this is STD screening. So they will use a chemiluminescent DNA probe that will look for the specific target sequences that are known to be found in chlamydia and known to be found in gonorrhea. When they find them, they'll join up and then they will luminesce. They will make a light. And that's how we know if somebody has chlamydia or somebody has gonorrhea. So instead of trying to grow it out on the agar plate, test it, they can do this much more quickly than we can. Because remember, in the lab world, we have to grow all those bacteria out, and it takes at least 1 day to grow the bacteria, at least one. Some bacteria take a few days. Some bacteria take 20-some days. It just depends on your bacteria that is going to be there. So it's not a fast, quick process in micro, but with these new molecular techniques like this is all done in an instrument, just looking for DNA. If the DNA sequence is there that they know is related to chlamydia, yeah, you have chlamydia. If it's not, it's negative. It's as simple as that. It doesn't take the day or days like it used to. All right, polymerase chain reaction, PCR. Hopefully you've heard of PCR before. Maybe you're not an expert, but I do have a video coming up as an announcement. I believe it comes out Wednesday. YouTube is great for lots of stuff. I know Khan Academy had a video on PCR that I thought was kind of good, but the one I put up is not Khan Academy, it's a different one. But it just really helps explain PCR very basic, what really goes on in PCR. So essentially what you're using PCR for is to make a ton of copies of DNA. So if you have DNA that you are wanting to make exponential copies of, you can easily do so in 30 to 40 cycles. So basically there's 3 steps in each cycle that occurs. And then once the cycle's done, it just repeats and repeats, making more and more and more DNA. So the first step is it will denature. It will separate the double strands. So you have 2 different strands. It will add on primers to the target. And then it will use DNA polymerase enzyme to make new strands of DNA, essentially. So in micro, we are using that PCR to diagnose bacteria, viruses, fungus, parasites, so all of our microbes. We're also heavily using it for looking at antibiotic-resistant bacteria. We are using it for key viruses like HIV, hepatitis C. So there's one type of molecular instrument that I put there. It's called the Cephia Gene Expert. I have seen the Gene Expert in different labs that I visited here in Phoenix. It's a big one that a lot of micro labs are picking up. And, well, if you know, Phoenix is a huge city. It's like 4 million people plus. So obviously they have a huge population. So these are large micro labs. These are not small labs. So, of course, they have the latest and biggest instrumentation. For me, it's kind of nice because I come from Fargo, and I'm like, oh my gosh, I didn't get to see this there. So, but yes, they have the Gene Experts, and so these are different things that they will look for that run on the Gene Expert. So you have your methicillin-resistant Staph aureus, C. difficile, norovirus, Ebola, Group B strep, tuberculosis in 2 hours, and there's many, many more. The list went on and on. I just highlighted some. Now, the tuberculosis in 2 hours is crazy. Mycobacterium tuberculosis, which is, there's a couple different ones that cause tuberculosis, but they all are very slow growers. They literally take way longer than 7 days to grow on an agar plate. So you're looking at somebody who's coming in, potentially having tuberculosis, which is a very serious disease, and we can't even diagnose them with tuberculosis because we got to wait for the bugger to grow out for over 7 days. I mean, that's crazy. And now, just through using genes and PCR and DNA, they can tell the doctor yay or nay within 2 hours. That's awesome. Like, it's insane when I think of the difference between what it used to be and what it's becoming. All right, so the future of molecular diagnostics and micro, hopefully as molecular testing gets to be more common, more just normal to see, hopefully it'll get easier to use for people, cheaper. It's definitely already more efficient, more accurate. And it also allows for a lot more standardization between labs. The thing with micro right now is micro's always been a very heavily hands-on testing type of department. And you look at the colony, you kind of figure, okay, I think it might be this. You start doing all these little tests that you can do on the bench yourself. You know, it was very suggestible to change or differences between tech to tech. Like, I might run one of those little tests differently, or I might decide, no, I want to do this test or this test. It was very subjective. And with PCR, you throw all of it in there, it does it, and it spits it back out so fast. And it's standard. It's a standardized process instead of it being, hopefully that tech is trained in right and knowing what steps to use and not going to proper order or something. So I know, I feel like I'm blabbing. Hopefully you get it. Talk to your lab instructors. I think it'll be fun to hear their take on everything and what they've seen and all that. I did pull up a PDF file here. This is the gene expert that we kind of talked about. Here's a bunch of the things that it does. Again, 35-minute screening for group B strep in labor delivery. That's huge. Again, it would take us a minimum 1 to 2 days, 1 day to grow up group B strep, another day to identify it. So it would normally take 2 days to tell them, yeah, you did have this. Now it's 35 minutes. So it's, again, 2-hour detection of tuberculosis. That's crazy. 45-minute detection of C. diff. So yeah, there's a lot of extremely efficient things here, and it really helps us care for those patients better. It helps us make sure they get the right, you know, right therapy, the right antibiotics, especially the drug-resistant ones. We can make sure we definitely give them the right antibiotics if they're resistant to other drugs. We can make sure they get into isolation. If they're in the hospital, we need to make sure they're in certain isolation if they have certain diseases. Like if they have C. diff, Clostridium difficile, they have to be in contact isolation. They can't be just sitting with anybody. So, you know, that's where this is really helpful. Okay, so that's enough of me talking. That's it for this week. Again, these 2 chapters are not really tested on in our class. It's just information I wanted you to know that's up and coming and to be aware of. So if you guys have any questions, just let me know or ask your lab instructors. They're amazing. Otherwise, have a fabulous week. And again, you do have your first quiz this week over material from weeks 1 and 2. It's heavily concentrated on week 2 with all the augers and the stains and that kind of thing. All right, good luck and have a fabulous one.